for making it and staying until the very last day. I'm Kensani Nkuna, and I'm going to be chairing this session. We have an exciting lineup of presentation ahead, focusing on disturbance and soil, and plants, sorry. I'd like to remind the speakers to be mindful of their time and leave room for questions if they wish to have some, and also to just try and have a smooth transition between the speakers. So if you know you're speaking next, so just come and sit on the chairs right in front so we can move swiftly. So as you know, you have an option to get mic'd up, and if you want to be mobile. If you want to do that, just be mindful of the light there and the screen. Just don't trip yourself over. Yeah, I guess without further ado, we're going to kick off this session with Andrew's 5 minutes presentation. Take it away, Andrew. Thanks.
Good morning. Thank you all for coming. It's much appreciated as the first session of the last day. So I'm going to talk a little bit about zoogeochemistry and wildlife management. So I'm sure a few of you in the room are probably thinking, what is zoogeochemistry and why is it important? So it's been known for a long time that life needs certain chemical elements to thrive. And whether it's able to access those elements influences what life exists, how productive that life is, and also some ecosystem properties such as things like carbon storage. But animals aren't passive to the chemical landscapes within which they live. And by being able to move, they can actively reshape the chemistry of their environments. And they may do this through ways such as the creation of hotspots. As an example, you may see something like a carcass site. By actively picking up elements and dispersing them across the landscape in both their excreta and their bodies. Or they may also interact with other ecosystem processes, such as things like fire. But as with all things, humans manage to get in the way and mess up a lot of these natural processes. And principally, they do this by changing what animals are in the environment, by changing what elements these animals interact with, and also by changing how animals move across their environments. And to demonstrate some of these, I'm just going to quickly run through a few examples from a place I've been working for a long time, which is Swallow Kalahari Reserve. And you'll notice that the reserve is split into two sections. One section which is principally has cheetah, wild dog, and a few spotted hyena, and the other section which is dominated by lions, which I'll come on to as to why that's important. And Swallow is just one example, and just to really sort of show that we make a lot of management decisions, but we don't necessarily know the cascading effects that may come from this. So first of all, managers to some degree always influence what animals exist in the landscape. At Swallow, they reintroduced a number of native animals that had been removed during farmland and brought them back into the landscape. But which of those animal populations do well will often come down to different sorts of management styles. So in the section where the lions are present, the blue line, springbok numbers have been increasing because lions don't particularly go for springbok. But where there's been high levels of cheetah predation, springbok numbers have crashed quite substantially. And then there's sometimes some subtle ways in which our management styles might influence where these nutrients end up. So scavengers pick nutrients up from carcass sites, and you probably have been driving along roads in Kruger and seen these hyena feces, these very obvious white hyena feces. And these are super rich in things like phosphorus and calcium, but hyena tend to communicate via olfactory communication, so they defecate at intersections of roads. So where we put our roads will often mean that nutrients that come from a carcass may end up specifically in certain parts of the landscape because of our practices. And in certain cases, predators aren't able to regulate herbivores enough, in which case management will often remove animals from a reserve to prevent things like overgrazing. And in Swallow, they've sometimes moved up to 1,000 animals per year. And this leaching of nutrients from the environment over time may be substantial. And we found, doing some back-of-the-envelope calculations, that the nutrient losses from this process may be as much as is being input from atmospheric deposition. Within the site, Swallow try to help out some of the animals with a bit of animal husbandry, and they provide 10,000 kilograms of salt licks and 25,000 kilograms of mineral licks. And these really shape how animals move. And using camera trap information, we're able to find that it's particularly the largest animals that are the ones that are using these licks. So adding this all together, if management is really influencing a lot of these natural processes, could we actively use management to try and achieve certain conservation goals? So that might be the protection or conservation of certain species. So the black rhino is a species that is in conservation crisis, and but it also has a particularly high salt demand. Adding salt licks in swallow is almost certainly helping out this species, as it's a very low sodium environment. However, there's a litany of problems or changes that occur when you put salt licks in the environment. This is a site of animal interaction, you'll see here, between the rhino and the giraffe, but it also starts to influence things like seed dispersal, localized sites of high vegetation impact, and also we start changing the quality and the quantity of animal feces, which is going to be important for things like coprophagous beetles. So if we do one action, we may have consequences elsewhere. And the final thing which I'll finish with is that animals are increasingly being promoted to be important for things like climate change. And I would say that this is really important, but we should make sure that we don't overly focus on the role of animals in being able to sequester carbon. This is principally because there's a lot of high uncertainty with a lot of these studies still. A lot of these processes may take time, and we also start to create a lot of ethical dilemmas depending on how we value the wildlife we have in our systems. How do we think of a savanna elephant if we start realizing that it emits carbon by knocking over trees? Instead, if we are going to manage for zoogeochemical processes, I suggest that we should do ones which increase the complexity and the connectivity of chemical landscapes, because this really helps more resilient ecosystems, which is what we really need right now. And with that, thank you very much.
Thank you. You've used up your time. Our next speaker is Celeste, and she's going to talk about the influence of large mammal grazing on grassroot biomass. Thank you.
Hi everyone, thank you for being here. I'm just going to tell you a little bit about how grazing influences the biomass of grasses. So we all know savanna systems are important. They sustain a host of wildlife species and perform a number of functions that are important to us humans. Grasses are an important component of savanna systems and influence a lot of these processes, such as carbon sequestration. And in fact, in tropical savannas, most of the carbon in soil comes from grasses. So root biomass is an important component and contributor to the carbon in soil. So we wanted to know, how does grazing influence the below-ground biomass of grasses? So grazing by wildlife can influence the below-ground biomass of grasses in different ways. So they can shift the species composition from perhaps a system that has a lot more bunch grasses, which are tall-growing grasses with vertical growing roots, to maybe a system that's more grazing adapted, where they have lawn grasses, which are these short, mat-forming grasses with a different root structure. So to see what this looks like, we went to Mfolozi Park, which is the oldest proclaimed national park in Africa. It was proclaimed in 1895. So it has a system that has been open to large grazers for about 127 years. And recently, in 2009, they've added sections to the park, which then opened up new areas to wild grazers. So taking advantage of this natural grazing gradient, we went and sampled using the park as an area with a long history of wildlife grazing, the new areas as having about 3 years of natural grazing, and using the community as a control. But of course, there are low levels of livestock grazing outside in the community areas. So from above ground surveys, what we found is that in the area with a longer history of wildlife grazing, we did find that most of the grasses that we found were lawn grasses, significantly so, more than in the areas with less grazing. There was slightly more lawn grasses in the community area, although this was not significant between the two areas that has low grazing. And then when looking at the community composition, you see the same thing. In the area where there's been grazing for a long time, the dominant species are the lawn grasses. So you have Iroquois, Mozambicensis, Panica maximin, Sporobolus, eucladus. And for the other two sites, you have a similar species composition, which is dominated by bunch grasses, Banica maximum, Themida triandra, Digitaria eriantha. And then when you put this in a PCA, you see the same thing where the area with a lot more grazing separates out and a lot more overlap in the areas with less grazing. So using these species would make up more than 75% of the abundance. We targeted them for below ground biomass extraction. So we would put plots of 30 centimeters by 30 centimeters and dug down to 10 centimeters and spend a lot of time washing and sorting roots. And what we found was that in the area with less grazing, there was a higher grass biomass compared to the area with lower grazing. Unfortunately, we were unable to sample in the community area due to some external factors, so we couldn't extract roots from there. But something interesting that we did found, when you look at how grass biomass relates to, the root biomass relates to grass cover, with the lawn grasses, regardless of the grass cover, you have more or less the same root biomass. This means that whether you have 35% bare ground or 10% bare ground, you have more or less the same amount of root below ground. This is not the same for bunch grasses, where the amount of root biomass increases with grass cover. Then when looking at root to shoot ratios, it's not a surprise to then see that in the areas with more grazing, you have higher root to shoot ratios compared to the areas with less grazing. And this reflects the difference in the species composition, with a lot more lawn grasses in the area with more grazing compared to the others. And again, unfortunately, we can't compare this to the community area because we weren't able to sample there. So this shows how grazing can influence below ground biomass at the community level. But there can also be other mechanisms that influences the below ground biomass. So what I'm doing now is I'm looking at how individuals of specific species responds to grazing. And I'm doing this currently in the park. So if anyone's interested, please come and speak to me. I'll be happy to tell you more about it and to maybe even go show you. So we have different functional grass types, and we have some treatments where they're not being clipped, some where they're being clipped at a low intensity, and some where they're being clipped at high intensity. So I think there's a lot to unpack, there's a lot going on, and there's a lot of things that we can still look at with this below ground biomass. So please come and talk to me, because I can't go into it in 5 minutes. Thank you.
Thank you, Celeste. We've run out of time. Great job. Our next speaker is Helga, and she's going to be giving us a 15-minute talk about the distinctions between megacarcass site and the surrounding matrix.
Good morning, everyone. So, decomposing large vertebrate carcasses are an inherent natural disturbance of ecosystems. They are a localized source of nutrient enrichment, and they have strong impacts on ecosystem biogeochemistry and also the community dynamics of vertebrates, invertebrates, microbiota, and vascular plants. And although I will be presenting on some vegetation dynamics today, this forms part of a larger ecological question, because we have limited knowledge on the individual carcass effects on spatiotemporal heterogeneity in terrestrial ecosystem processes. And surprisingly, there's a much clearer understanding for marine ecosystems, where whale megacarcasses create these oases of biodiversity and production that may persist for decades. So, despite being the largest land animal, we know little about how the nutrients from the megacarcasses of elephants affect savanna ecosystems. And given that elephant mortality is a natural part of the savanna, and that annual mortality of elephants may total several hundred individuals annually, the nutrient pulses of about 800 kilograms of carbon, 300 kilograms of nitrogen, and 125 kilograms of phosphorus for a single 4-ton individual, adult individual, and their impacts are likely to operate at the landscape scale. So these nutrient pulses often create nutrient hotspots, and given the sheer size of elephants and their ability to persist in the environment for a long time, they are likely going to cause long-lasting legacy effects on the environment and the processes. And these carcass-derived nutrients have been found to cause shifts in plant communities. So we therefore anticipated that carcass plots will be dominated by a unique set of species and traits, likely adapted to high fertility soil. So I will be presenting some vegetation data that we sampled on megacarcasses of 10 elephants on granitic and basaltic soils in the Kruger National Park. So at each carcass site, we established 4 transects of 15 meters each in the 4 cardinal directions. Vegetation was sampled in 1 meter squared subplots placed at various distances from the carcasses. As you can see, at 1 meter, 2.5 meters, 5, 10, and 14 meters. And within these 1 meter squared subplots, we identified all life forms, and we also recorded total percentage cover of vegetation, bare soil, rock, litter, wood, and dung. So we considered carcass sites to be 0 to 3.5 meters from the carcass, and matrix sites were considered to be 10 meters and further away. Now, this was based on field observations where we could clearly see in the field a clear zone of impact, and these field observations were then corroborated by preliminary soil results, where we found that soil nitrogen and phosphorus was much higher at carcass sites. Similarly, other nutrients such as sodium, manganese, and zinc was also higher in the soil at carcass sites. And then also, we looked at some dominant grass species such as Eurocloa mosambicensis, Cynodon-dactylon, and Panica maximum, and they had higher levels of sodium, iron, and aluminum at carcass sites compared to matrix sites. But Courtney will tell you a little bit more about that. So we found that the species composition differed significantly between carcass and matrix sites, and we also didn't find any significant variance in dispersion, so we can confidently say that these communities differed significantly. And these significant differences in plant community assemblages is likely due to the differences in soil nutrient concentration. And these increased nutrients at the carcass sites leads to the formation of cadaver decomposition islands, and they are these nutrient-rich bare patches which facilitate the recruitment of species that could otherwise not have established there, and they also support vigorous plant growth during secondary succession. Interestingly, the changes in species composition between carcass and matrix sites were mainly driven by grasses, and the higher levels of soil nitrogen and phosphorus at the carcass sites led to the formation of biogeochemical hotspots, and these are predisposed to form grazing lawns. And the inherent increased herbivore activity helps maintain high productivity of grasses. And I just want to draw your attention that we had carcasses that was from 0 to 2 years old, and I think a lot of them might have been a bit older, so maybe we were more in this stage of succession. But if we were to sample more fresher sites, patterns might be very different, so yeah, just to keep that in mind. So species that contributed most to the significant differences between carcass and matrix sites that dominated carcass sites were all palatable species or species of high forage quality, and this is to be expected at nutrient hotspots. 80% of these species were perennial grasses, although we might have expected more annual species because of the high level of grazing and disturbance at these sites. And although we know that forbs tend to perform better with improved soil conditions, grasses tended to dominate these sites. And then 60% of the species that dominated carcass sites had a prostrate growth form, which is a functional adaptation for spatial refuge from grazing, and they also have the potential to provide nutritious and high bulk density forage for herbivores. When looking at the matrix sites, dominant species were mainly perennial tufted grasses, and maybe the lack of high forb abundances here could be due to competitive exclusion effects, where these tall, fast-growing tufted grasses outcompete forbs of a lower stature for resources such as water, light, and nutrients. And then also the high levels of productivity, together with lower levels of grazing a bit further away from the carcass, might have favored the establishment of tufted grasses with high leaf production. And then interestingly, most of these species were also palatable, and this supports the notion that areas surrounding these mega carcasses become zones of fertility, and then favoring the establishment of high quality forage. So this study definitely contributes to a growing recognition that megacarcasses have significant effects on terrestrial ecosystems. And the effects of elephant carcasses on semi-arid herbaceous vegetation structure and function, demonstrated here for the first time, expands this awareness significantly, given that elephant mortality is a natural phenomenon in the Kruger National Park. So megacarcasses therefore represent a unique disturbance type, adding to the complexity of semi-arid savanna systems by creating these nutrient hotspots with distinct floristics and then also increased forage potential. So what I would like you to take home is that elephant carcasses appear to play an important ecological role in the terrestrial ecosystems of semi-arid savannas. We saw that they influence herbaceous plant community composition and structure because carcass sites were floristically and structurally different from the surrounding matrix. And although in this case the floristic changes were mainly driven by grasses, they definitely deserve more attention to see the responses of other life forms. And this is where the time component comes in, to remember that over time, what we might expect to happen will change. And then lastly, megacarcass sites are considered nutrient hotspots, supporting mainly palatable species, and they are therefore considered functionally important for the semi-arid savanna landscape. And I feel like I really ran through this, but yeah, this is a really fun project to be a part. We are a multidisciplinary, multi-institutional group of people looking at soil and insects and large herbivores and bones. And yeah, we are staying behind for some fieldwork, so we are still collecting some data. And then hopefully next year I can come and tell you if that temporal aspect does have an effect on the patterns that I presented today. Thank you so much.
Thank you, Helga. We have a little bit of time to take a few questions. Can I have a mic there? I'll start by Ben.
Thanks, Helga, for a great talk. I was just wondering, you said you did sample on granites and basalts, and whether you picked up differences, or like on the nutrient-poor granites, did you have a bigger effect? Or was the boost of nutrients so high it overrid even on the basalts?
Yeah, so thanks, Ben. Like I said, yeah, I kind of pooled all the data in terms of age and soil type, so I haven't really teased out the differences in soil, but yeah, that is one of the next steps that we are going to look at.
Ladies first.
Lawrence, thank you. I think that is what we are trying to figure out now. But yeah, I think it might be long. I think these nutrient hotspots might last for a long time, maybe also even decades, such as the whales, but we'll wait and see.
Hi, thank you very much. That was very interesting. I'm particularly interested in this temporal component of the development of the nutrient hotspot. So not only that there are more nutrients, but the nutrients and the combination, the stoichiometric ratios of these nutrients should also change over time, right? Because at first you just have this fleshy component, and after a while when the bones start to decay and so on, you might have a lot more phosphorus and a lot more calcium coming in. So are you at all going to check on like the kind of the stoichiometric development of that landscape and how that might open up kind of nutrient niche space for different species?
Yes, thank you for that. Definitely, we are, like I said, a big team of people, and there are people interested in that particularly. So I actually hope that the project might be extended a little bit because we have so many questions that we want to answer, and that is definitely one of them.
Okay, thank you. Thank you, Helga. Great job. Up next is Courtney Reed, and she's going to continue on the topic of elephant carcasses, looking at soil and plant nutrients.
Hi, everyone. Thanks to Helga for introducing everyone to our project, and I'm really excited to tell you more about it. Before we jump in, just want to go ahead and thank our group of collaborators, the kind of megacarcass ecology working group, and then the institutions that are supporting our work. So we know that elephants have these really substantial impacts on savannas to the point where we're always calling them ecosystem engineers. But we don't actually know how that plays out after they've already died. Are they still engineering ecosystems just with the size and the impact of their carcasses? And so that's what I'm really excited to find out. And so about 300 elephants die each year in Kruger, and that's a huge influx of nutrients into the savanna from each of these carcasses. So just to put it kind of in context for you, the amount of nitrogen you get from one elephant carcass, just like an average-sized elephant carcass, is the same amount that you would get on a full hectare of savanna for 100 years of atmospheric deposition. So that is a huge amount of nutrients all coming in at one spot at these hundreds of locations across the park each year, and could potentially have enormous impacts, especially on nitrogen-limited soils. So we have three kind of main hypotheses I'm going to talk about today for how these nutrients from carcasses are affecting soils and plants. The first is that nutrients will move from carcasses into the soil, and we're especially looking at kind of phosphorus, nitrogen, and carbon initially. Second is that influx of carbon would stimulate soil microbial activity, and you'd have increases in soil respiration. And then the third is that kind of over time, those nutrients would move from soil into plants and then be available in these above ground nutrient pools. So to test this, we so far have looked at 10 carcass sites between 0 and 2 years post-death and on a split between granitic and basaltic soils. So at each of those sites, we ran transects in the 4 cardinal directions, and we took soil and leaf samples at 0, 2.5, 5, 10, and 15 meters. The plants were leaves from Eurocloa. But at each of those, we pooled the samples, so we had 1 leaf sample and 1 soil sample at each distance per site. And we analyzed them for nutrient content. So what we found first is that soil nitrogen and phosphorus are both elevated at carcass sites. So if you look here on the x-axis, you kind of get further away from the carcass as you move along the x-axis than soil ammonium on the y. Basalt soil is blue and granite soil is yellow. And you can see that especially in the granite soil, there's a decrease in soil ammonium with distance. Interestingly, not with nitrate yet, just with ammonium, so that conversion hasn't really happened in the 2-year time span. We see the same thing with phosphorus, especially in the granite soil. We have this significant interaction where soil phosphorus decreases as you move away from the carcass. And we know that this increased nitrogen is actually coming from the carcasses because we've done our isotopic analyses showing that the proportion of nitrogen in the soil is actually coming from those carcasses. You can see that's true in both soil types. The proportion of nitrogen in the soil from animal sources is greater underneath the carcass sites. For our second hypothesis, we found that soil respiration and soil carbon are positively correlated. It's a good gut check. But then also that both of these are higher closer to the carcass site and lower as you get further away. And what's really exciting is that these really high sites are actually from the center of the carcass at our youngest site that was only 24 days post-death. So we're really excited to get some that greater temporal span that Helga was talking about and have a better understanding of those initial dynamics. And finally, we found that leaf nitrogen is elevated at carcass sites. So this is leaf percent nitrogen. You can see decreases with distance, but also leaf isotopic nitrogen, indicating again that it's actually coming from the carcasses and moving up into those leaves. So just to recap, we've just examined the impacts of elephant carcasses on soil and plant nutrients. We have found that the nutrients move from elephant carcasses into the soil, that carbon from those carcasses increases soil respiration. These nutrients move from soil into plants, making them available in these above-ground nutrient pools. And finally, we're just really excited because it's showing us this kind of first initial evidence that these elephant carcasses really do have substantial impacts on the savanna and create these nutrient hotspots that we're excited to follow for years to come. Thank you.
Thank you, Courtney. Moving on, we have another 15 minutes talk by Michael, and he's going to discuss the long-term change in the Serengeti ecosystems.
Yeah, thank you. Thanks to Sand Parks. Happy to be here. Great to interact with a lot of friends and colleagues again. If you haven't heard the news, I'm here to tell you that my elephants are stronger than your elephants. And I know that because it says it right up there. No, but seriously, if you're an elephant biologist and you saw this title and got annoyed and you wanted to come here and see what is this plant ecologist going to be talking about, then mission accomplished. I'm trying to convince you today that this is a talk about elephants, but really it's about trees. I'd like to start by thanking some of our collaborators and research assistants, Jeremiah Sarakikia, Reginald Sukumis collected a lot of data for this project. Two great grad students, Dan Griffith and Deostidith Rugamalila. Two amazing postdocs, Tom Morrison and Jason Donaldson, who's here in the audience. Thanks for all your work on this. And then my long-term collaborator, Ricardo Holdo at the University of Georgia. So from this really well-known paper by Mahesh Sankaran, we know that rainfall sets an upper bound to tree cover across African savannas. We also know that this has been a major savanna research question for essentially the last decade, and especially what controls variation under this curve. From some work by Gareth Hempson and Sally Archibald, we know that across this precipitation gradient, the two major consumers, fire and herbivores, have very different effects, with fire dominating at high rainfall sites and grazers and browsers dominating at intermediate or lower rainfall sites. So this sets up the scenario where you may have an intermediate site, such as this one, dominated by grazers. They consume a lot of grass biomass. This suppresses fire, which encourages tree growth, and so you can have high tree occupancy despite having a lot of grazers and browsers. There's another scenario here where these points at high rainfall and low tree cover may occur because there's a lot of grass biomass which is burned and consumed by that intense fire, which tends to kill tree seedlings. Kill tree seedlings. Now there's another really important agent of disturbance in these systems, which is demonstrated by these photos. So these are some camera trap photos at one of our long-term research sites, and you'll see what happens here. So along comes one of these male elephants and takes out this tree in less than, well, essentially 1 minute. This is a 40 to 60-year-old acacia tortoise, had been growing in this savanna site, and within a minute is able to take it out and knock it over. So obviously these elephants are really important agents of disturbance, and we need to think about them when we're trying to understand woody cover across savannas. And given their large ranges and large dispersal capabilities, elephants really exert an influence across this entire range. Now, there's been some excellent work done on this, some really nice meta-analysis of elephant impacts across African savannas. This one in particular reviewed 238 different papers and showed that 130 of these different studies showed negative impacts of elephants, 65 had no effect of elephants, and 43 actually had positive effects of elephants on woody cover. They went on further to explore the effects of rainfall, and what they found is that at arid sites, there were always negative effects of increased elephants or increased elephant densities, whereas at transitional or mesic sites, there were variable effects, sometimes negative, sometimes positive. So rainfall seemed to play some sort of an effect in this, in mediating or modulating the effect of elephants on woody cover. Now, another really important and unanswered question, I think, is how these sites rebound, or how do they regrow? How does the tree cover increase after disturbance? How does this happen? So does it happen continuously through time? Does it happen episodically? And how fast do these transitions happen? I think this is something that we need long-term monitoring and long-term data to answer, and how fast these transitions occur. In the Serengeti ecosystem in particular, and you heard Craig Packer talk about this story the other day, these changes in woody cover and recruitment have been linked to this rinderpest outbreak that spread from Ethiopia in 1887 to the Cape of the continent in 1896. There was a catastrophic die-off of ruminants and cattle across the continent, and this included a massive decline in the Serengeti wildebeest. Rinderpest was eradicated, and then in the early 1960s, data showed that the wildebeest population in Serengeti began to increase and rebound. And as Craig talked about, I'm going to show exactly the same graph from Tony Sinclair. As the wildebeest numbers increased, the area burnt in the park, so the amount of fire in the park went down. And then the explanation is that this triggered a series of increases in woody cover, such as shown in these photographs. So this would have been really this scenario here, where grazers are suppressing grass, consuming grass, suppressing fire, and then encouraging tree growth. And I'll never forget, I think it was probably around 2007, I showed these photographs to William Bond, and he said, "Okay, but is this new recruitment or woody thickening?" And I had no idea what he was talking about, and only now, like 10 years later, do I understand how important his question was. What he meant are these new trees that are getting in via seed recruitment, or is this the increase in stems, existing stems, and these gullivers that really play this essential role in these systems? And I didn't know the answer. So in 2009, we set up these long-term study sites in Serengeti. We set up 36 monitoring plots at nine sites. And in these, we quantified recruitment, growth and survival of plants. We spatially mapped every woody plant and measured height, diameter, and damage. And then, importantly, we tracked the fate of individually marked trees and really tried to distinguish between top kill and true mortality. Okay, and that was a really important part of this study, and is really, you can't do it with LiDAR. You have to go out and go and visit these trees on an annual basis and see what they're doing on the ground. Okay, so I don't have time to talk about this part of the study, but we did it within the context of we excluded herbivory with cages, and then we burnt these plots. And this is a Google. I'm quite proud of this Google satellite image of our little exclusion plot there, showing we actually were able to keep fire out of these plots. We had weather stations across our study site. Again, I don't have time to go into some of these details, but what did we find? What did we find since 2009? What did we observe? Well, we observed, I would say, systematic reduction of tree cover by elephants. Every time we visited these plots, it was just like bulldozers, absolute catastrophe had taken place in these plots through time. Now, that probably doesn't surprise you, because everybody knows the impact of these elephants in these ecosystems. That's not the surprising part. But over this 14-year study, we just saw the elimination of tree cover. So I'm going to show you a couple of photos from 2010 and then again from 2023. These are just a couple of our research sites, just to demonstrate the significant changes that occurred. And then here's the last one. So you can really see visually the effect over time that these elephants were having. Now, we monitored and quantified the mortality on an individual tree basis, and we found that there was, on average, a 7% mortality rate per year. So across our sites, this is a map of Serengeti with our individual 9 sites, and each one of these bars is one of the 4 plots. The black bars are burned, and the gray bars are unburned sites. And what we found, and this is over a 4-year period, what was the proportion of total mortality? And you see there's just widespread mortality across all these sites, and there's really no interaction with fire whatsoever. It was, in fact, it was elephant damage, which was the single best. Elephant damage was the single best predictor of annual mortality. So this is like the individual effect of an elephant impact in terms of severity on a tree, and then the probability of survival. And for all the species, obviously there was a negative relationship, but it varied a lot among species. There was an 18.5% annual mortality for Vachellia senegal and only 1.5% for Balanites aegyptica. So really quite different effects across species. And looking at the plot level after 14 years, only 1 plot, so these are our sites across sort of a rainfall gradient map. Darker green areas are more rainfall, white or orange areas are lower rainfall. And these are the plot data over time. So if values occur above this 1 line, that means there were more trees in the plot than the starting number. And if it's below this, then there are fewer trees in the plot over time. And amazingly, there was 1 plot out of our 36 that actually increased in tree cover. A few of these plots had essentially no change, but 33 of the plots declined in tree cover. And so what was it about this plot that was interesting or unique? Well, so we were out in just this last year in 2023, and after resurveying this plot, we learned that it was increasing stem number and the diameter of existing trees that led to this increase. It was not new woody recruitment. It was not new individuals. So even after 14 years, the only plot that increased in time did so via this woody thickening. The thing that I need to make you aware of is that the Serengeti elephant population has been increasing rapidly recently. So from 2006 to 2014, it went from just under 2,000 to over 6,000. And then the most recent count is somewhere around 7,000 elephants in Serengeti National Park. Okay, well, how is it? This raises a really interesting and important question. How is it with such a sort of a pathetic density of elephants, how is it that they have such catastrophic effects? So if you look across a histogram of elephant densities in Africa, shown here, I mean, Serengeti's way down here. It's kind of in this like meager elephant density. So how is it, even with the increasing numbers, the density is only about 0.28 elephants per kilometer squared. So how do we explain this? Well, we propose 3 hypotheses to explain this. The first is that our elephants are just stronger than yours. They're just, they're super strong and they can just knock down a lot of trees. Well, the second one is that our trees are less well defended or more susceptible to elephant damage. And then the third is that poor soil growing conditions and strong grass competition reduces trees' capacity to escape and tolerate and recover from relatively moderate elephant damage. As I've already told you, this was just sort of a provocative title to get you to come here. Thank you very much. The second one, we don't really believe this because we have a lot of the same tree species. These are incredibly well defended. They have very thick bark. It just doesn't seem to be a good explanation, which leaves us with this explanation. And I'm just going to show you a couple of pieces of data that actually support this. So we have another study going on where we collected soils and looked at hydrology and resources across these woody to open gradients. And what we find is that there are areas of Serengeti where trees can literally not grow because of the physical constraints of the soils and the important effects of grass competition. So woodlands only occur in these well-drained soils with high water potentials, and grass competition is really limiting tree distributions. This final study was more of a simulation study, and what we did is we looked at tree escape probability based on existing woody cover. And at current levels of disturbance, which are this panel here, really you need like very high tree cover, 40% tree cover for new recruits to escape and get out of this, essentially trap, be it an elephant trap or a fire trap or whatever. And so we're sitting here, but as the number of elephants are increasing, it's really pushing us into this very high frequency of disturbance state, which is going to be even less and less chance for individual trees to escape. And to put it in even a broader context, this is from Caroline Lehman and Bill Hoffman and a couple other, and Sally and William. They first proposed that this was a really important effect for fire, that there's this fire escape threshold that really depends on underlying growth rates and underlying capabilities of the trees to grow quickly. Well, I'd say that we can do this same thing, but really think about it in terms of elephants. So what we saw probably is that in 2010, we were probably in here in terms of the time since top killed and the frequency of disturbance, and now we're moving this way. And if we're in a low growth rate environment because of strong tree competition, we're really trapped in this treeless environment down here, and it's going to be really interesting to see what happens in the coming years. And my point here and the take home, the thing I'd like you to leave here with, is that models that place emphasis on tree growth rates and soils, not rainfall, but growth rates of trees and soils as modulators, are really going to have the best chance for very broad application in this field. Now this is, I'm not the first to propose this. There are some really nice papers out there doing these kind of things, but I'd like to just put out there that I think a lot of us have the kind of data that we could bring together and do something really interesting on a sort of continental scale. So I'm just going to end by saying that if you're interested in this, we should put our soil and tree growth rate data together and elephant densities and try to come up with something more comprehensive. And with that, I thank you. I maybe don't have time for questions, but thanks very much.
Thank you, Michael. Our next speaker is Frank, and he's going to be talking about the survivorship and and the mortality of sycamore fig trees in the Kruger.
Now you can hear me. I think you could hear me before. Thanks, Sand Parks, for the privilege of working here for 2 decades. And from the early years, I want to remember our facilitators, Andrew Deacon, Patricia Cosa, Angela Gaylard, Sandra McFadgen, and more recently, Sam, Corley, Sharon, and Danny. Thank you so much. So what's taller than a giraffe, stronger than Michael's strongest elephant, and slower than a leopard tortoise? The mighty sycamore fig. You can see them in their brilliance in front of the Cattle Baron restaurant. These mighty trees can produce fruit crops up to 4 times a year per individual, 50,000 grams of wet biomass in fruit. They per fruiting cycle, and they're reputed to feed more species of birds and mammals throughout Africa than any other tree. Here's some of the biodiversity that I've photographed over the years right here in Kruger. Particularly important are 2 species of epauleted fruit bats, Epimaphorus, trumpeter hornbills. Those species are very good dispersers of seeds. Green fruit pigeons are tied heavily to the fruit crop for their food, but they grind the seeds in a gizzard and are actually seed predators. So the objective of our study early was tied on to foraging movements of the epauleted fruit bats, which we radio tracked and have published on. But now I want to talk about the distribution of the fig trees, the mortality survivorship since 2003, and gap distances between clusters. So 1,165 trees, each represented by a black dot, have been mapped and lodged the database here with scientific services. You can see that they're found from the Crocodile on up to the Luvuvu, Limpopo, all the major rivers and minor rivers as well. They're a riverbank specialist. They need to have their roots in the water. But of that original cohort of 865 trees that we mapped in the first year, 298 trees are now gone, mostly through the flooding events of the last decade or so. If the flood of the century occurs once a century or maybe twice a century, it's good for recruitment of figs. The wood piles of debris created up on the riverbanks by floods are ideal places for young seedlings to grow, receive nutrients, and maybe some protection from browsing and trampling. But when the flood of the century occurs every 2 to 3 to 4 years, it's disastrous, and there's virtually no recruitment to replace the trees that have been lost in the last 20 years. We found a total of 3 individuals that I would say have been recruited into the canopy or subcanopy population in our 20 years of study. Some rivers, like the Red Rocks region, maybe that's unique, the Red Rocks Upper Xingwezi are unique in that not a single fig has been lost from 2003. But the Mfangolo and the Xingwezi Kanidud section, the lower floodplains, which have received the brunt of quite a few extreme flood events, have lost 43% out of an original 149 trees mapped. I'm worried in particular about did I skip one? The center region below the Xingwezi, south of the Xingwezi down to the Olifants. Along the Olifants, 22 kilometers of river, 100% of the trees have been lost. The only blue dot there, surviving tree, is inside the camp up on the bluffs above the Olifants. Letaba camp is unique in that it had a lot of trees anthropomorphically planted in the camp, and those have been attacked, I think, by a combination of stresses from drought, lack of watering with water restrictions, and the polyphagous wood borer that's been invading recently. And I'm worried about the center region particularly. Here are some, the red lines and distances represent gaps between clusters, but in the central region, if we lose a few more fig trees, we will have an 80-kilometer gap between north and south, separating the dispersal agents, such as the bats, and the pollinators, such as fig wasps, and isolating the trees themselves into northern and southern populations. So our conclusion, 1 in 3 individuals of that original large data set are gone, virtually no recruitment. Fortunately, fruit production, the pollination system, the seed dispersal system, the viability of seed germination are all still functional. But for population stability, I think we may need to require some management in the future. Thank you.
Thank you, Frank. Our next speaker is Cassie, and she's going to talk about fire exclusion and early dry season, the negative impacts on plant communities.
Morning, everyone. I'm Cassie Rodrigues, and I will talk about fire exclusion and early dry season fires negatively impact plant communities of tropical savannas. Tropical savannas are influenced and maintained by fire. Fire removes a large amount of above-ground biomass, altering environmental conditions like availability of nutrients, light, and space, changing relationship among plant species. Thus, fire influences assembling plant community, which can impact their functionality. Fire regime describes the frequency and seasonal occurrence patterns of fire in a particular location. So fire regime can affect plant regeneration by its influence on fire behavior according to phenological and growth stage of plants during a fire event and due to post-fire conditions it imposes on plants, such as water restriction time. So changes in fire frequency and season can affect plant communities. Thus, we aim to compare the effect of different fire regimes on growth form cover, structure, and taxonomic and functional diversity of herbaceous layer in upper tropical savannas. For this, we select an open savanna area for fire experiments in central Brazil. We established plots of fire frequency treatments, annual fires, biennial fires, and fire exclusion since 2011, and plots of fire season treatments burned biennially early, mid, and late dry season. For each treatment, you have 4 plots of 30 by 30 meters, where you fix 10 subplots of 1 by 1 meter for vegetation service in rain and dry seasons during 2013 to 2022. You evaluated the effect of fire regime on vegetation over this time and at the end of this period. But here I will present only final data resulting from 9 years of fire experiments. To calculate functional diversity, you measure functional traits of fire response, growth form, branch architecture, leaf nutrients content, height, and type of underground organs in individuals of a more dominant species in each treatment. Now our finds. About community structure, bare soil percentage was lower in exclusion plots, and that biomass was higher in early and exclusion plots. Both are important factors in recruitment and persistence of species and community. Among growth form cover, only forbs were affected by fire regime, with exclusion plots showing the lower forb cover than other treatments. As species richness, forb richness was lower in exclusion plots, while graminoids and total richness were lower in early and exclusion plots. We found reduced taxonomic diversity in early and exclusion plots, and fire exclusion also led to reduced functional diversity, while annual fires promoted the highest functional diversity. Looking in more detail at species composition, we observed that annual, mid, and late treatments promoted a more balanced distribution of species abundance, with small and large grasses, shrubs, and even forbs among dominant species. In contrast, early plots had only large grasses and shrubs among dominant species, and exclusion plots had only 2 dominant species, and both were large grasses. Dominance of large grasses may prevent the recruitment and persistence of small grasses and shrubs and forbs in community, compromising the species diversity. So, in conclusion, fire exclusion had the most negative impact on plant communities, indicate that prescribed fire is essential not only for controlling fuel load, but also for preserving the diversity of tropical savannas. Early dry season fires, the most used for fire management Cerrados protected area, reduced the diversity of communities. And herbaceous layers of tropical savannas are resilient to annual fires. So understand each fire regime's positive and negative aspects is crucial to using it efficiently and ensure the preservation of tropical savannas. Thanks so much.
Thanks, Cassie. Up next is Marco, and he is asking whether fire-related traits can explain the distribution of woody species across the Cerrado and the Atlantic rainforest.
Thank you very much. Hi everyone, my name is Marco. I'm a PG student from Brazil. I'm very happy to be back here Last time I got the opportunity to come was 2020, and then the pandemic hit. A lot of stuff happened, so being here today means a lot for me. And I'd like to thank everyone that's present here and everyone that made this happen. Today I'm going to talk about how fire-related traits can help us understanding some of the distribution of wood species across the Cerrado and the Atlantic rainforest. There are two hotspots for biodiversity in Brazil. So we know that savannas and forests that can coexist in the same area, many times occurring under the same climatic variables and under the same climate. Some areas that are currently harboring forests, they can also harbor savannas and vice versa. And this is the case of what seems to happen here in South America in some areas. So if we look specifically to the Cerrado and to the Atlantic rainforest, we have very different vegetation domains because the Cerrado is a mainly fire-prone vegetation type composed of great amount of savannas covering its landscape, while the Atlantic rainforest is mainly fire-sensitive. And despite these differences, we do have fire-sensitive vegetation types in the Cerrado, such as riparian forests, and we do have some flammable vegetation types in the Atlantic rainforest, like some grasslands. So this is some indicative that both the climate and disturbance may act together across these two major vegetation domains. This has been intriguing ecologists for a long time, and two key perspectives were drawn considering the distribution of forests and savannas in South America. And the first perspective, it's related to positive feedbacks. So some studies have been arguing that this different coexistence is mainly maintained by disturbance, such as fire, for instance, and land use and history. So the feedback between the vegetation and the disturbance would be responsible for maintaining this different biomes coexistence. The other perspective is more determinist and have been arguing that climate or soil properties would be determining the type of biome that you will find across these different landscapes. These two different perspectives, they are very reasonable, and both of them, they have this each importance, because there's a close relationship if we look to the evolution and how the Cerrado and Atlantic rainforest is distributed across the Brazilian territory. There's a close relationship between these two domains. So for instance, if we look for species from the Cerrado, we know that the core lineages, such Mimosa, Andira, Lupinos, and Microlici, they have its diversification from lineages that evolve in fire-free vegetation types, which is also observed in species from the genera Eriotheca and Paquira. And this evolution of these different core lineages, the plants that belong to these lineages and that have fire-adapted traits, they start to have their hybridization approximately 4 million years ago, which is when we do have grasses expanding, flammable grasses expanding in the core area of Brazil, and so a flammable ecosystem was born. So this is a strong indicative that maybe fire disturbance, as well as helping species to diversify, may be acting as a driver of savanna vegetation type where Atlantic forest and the Cerrado is occurring. But if we look to the determinist perspective, it also makes sense because the climate, the topography of the Atlantic forest is different from the Cerrado. So the vegetation from the coast is exposed to a different climate from the vegetation from the inland. So the determinist perspective can also make sense and bring some light in this coexistence. So based on these different perspectives, we've been asking, can the possible distribution of savanna and forest species explained by the climate also be explained by the species' fire-related traits? Or in other words, can we understand what the determinist perspective is saying to us from the positive feedback lenses? And to do that, we start looking at species from savannas and forests instead of looking to the forest and the savanna structural patterns and distribution per se. And we choose two very important traits found in wood species from savannas that allow them to survive fire, which was recently related to communal assembly across vegetation types from the Atlantic rainforest and from the Cerrado. So specifically, we've been looking at their aerial bud protection and to the rates of bark growth, because together these two traits are responsible for allowing species to resprout new branches after fire disturbance. We do know that species from forests, they show low or no bud protection, so their buds are very exposed to the environment. And species from savannas, they have medium to high bud protection, so they have a greater chance of surviving fire events and thus resprouting. The same idea applies for the rates of bark growth, and we've been observing that species from savannas, they can produce up to 2.5 times faster bark than species from forests, so it's another important trait. Not many species had these traits described, and we managed to work with 84 species from savannas and forests, and to make sure that our vision wouldn't be biased by considering only forest species or savanna species, we were lucky enough to have these traits described in a fairly equally proportion. So we managed to find 33 savanna species, 27 forest species, and 26 species that can grow into both types of environments. Okay, so this was the first step, the traits. We then, for each one of these 84 species, we retrieve their occurrence using the GBIF database. And we perform some process to analyze their suitability across different types of climates, across these two major domains. So we consider the areas where the species were sampled, here represented in ones. We created a background where the plants were not sampled, and we compared this raster file, this background, with 18 bioclimatic variables from the World Clim dataset, like temperature, rainfall, dry season length, etc. And then using the max-ent model, the max entropy, we assessed the possible distribution, or better, the places where this vegetation have a great suitability based on these climate variables. And then we validated using the Live-On group out to make sure that we were not biased by few records or few species being analyzing one or another vegetation type. The next stuff we did, we reconstructed the fire frequency from the Cerrado and the Atlantic rainforest from 1984 to 2022. And to do that, we used the data from Map Biomes, which is a great platform from Brazil, taking care of our land cover and use. And we calculated the number of fire events in each one of these pixels, and we segregated them into fire frequencies. So no fire, low, between 1 and 5 fire events, medium, 6 and 15, high, 16 and 25, and extreme, 26 to 32 fire events. The first step that we took, we considered the possible suitability that our models show us through the max entropy, and then we perform an overlap comparison with the fire frequency. So we considered this area of each distribution within the Cerrado or the Atlantic rainforest as species abundance, and then we calibrate these possible areas of distribution using the traits. So we assess the community weight means for each trait of each species across each one of these different vegetation types, considering fire disturbance. And what we got? First thing, we noticed that the more frequent fire events are within the possible area of suitability explained by the climate, the more important are the traits related to fire disturbance. So if you note here, areas with high to extreme fire disturbance, bud protection and bark growth rate are almost twice more important than areas with no or low fire events. So in other words, what the possible suitability explained by the climate it's telling us can also be understood through the lenses of the protection against fire, which would be related to the positive feedback perspective. If we start to look at how this explains the possible distribution, the possible suitability of species according to the climate across these different domains, what we see first is that in areas with no fire events, both bark growth rate and bud protection, both traits, they have lower values as the area with no fire increases under these different domains. But from the point that we start addressing and considering areas that have fire disturbance, an inverse trend is observed. So in areas from the Cerrado, bark growth rate shows a greater value as the proportion of the realm burns, and the same occurs for bud protection, but the inverse occurs for the Atlantic rainforest, which is a mainly fire-free vegetation type. So bark growth rate and bud protection, they have lower values and they are less important in these areas. We observe the same pattern in areas with medium fire events, medium frequency, 6 to 15 fire events. The same pattern for high fire frequencies and extreme fire frequencies, but with much lower data to rely on because not much of the territory can be burned 32 times in 36 years, so that's a very high frequency. So coming back to the question, can fire-related traits explain the suitability of species that were assessed based on the climate? This is very preliminary, but we believe that yes, that our results suggest that fire-related traits, they provide a complementary explanation to what the climate has been telling us. And that makes a question. If the savanna fire distribution is only determinist, only the determinist perspective can explain the distribution, then why do species have such well-adapted traits to the fire disturbance, which would be understood as something happening because of the positive feedback relationship between the vegetation and the disturbance? And this has been said for a long time, and especially from scientists that attended and keep attending this meeting, that maybe we should be moving towards an integrative perspective between the determinist perspective and the positive feedback perspectives. Alone, they may not suffice the idea of understanding what's going on in these areas. So these are very preliminary results. We do have some drawbacks. The first of one is that species that we use that had their traits described, they are from the southeastern part of this ecotone here between the Atlantic rainforest and the Cerrado. And this is an area with a lot of human population, so we have lower cover of vegetation, and therefore we have a lower fire frequency when compared to the core area of the Cerrado here. So we've been underestimating species and fire frequencies in this model so far. We only consider the above ground protection of the species against fire, but we do know that below ground traits are incredibly important for species in flammable ecosystems, but we did not counted these traits in our models yet, so we are digging some stuff so we can address the type, the potential of respawning from the below ground of these different species across these two different realms. And the determinist vision is not only based on the climate, as I said in the beginning, it can also be related to the topography, to the soil properties, the adaptive conditions, water table, and so on. So now we are also considering these variables in these models of possible suitability, and we are actually calculating the distribution and not just the possible suitability of species, which is a little more complicated than that. But despite of all of this, despite lower fire frequencies, despite traits that are incredibly related to fires not being considered, we do believe that we bring some indicative that the positive feedback perspective must be integrated to the determinist perspective when understand the coexistence of savannas and forest biomes. So thank you very much for your attention. That's what I got today.
Thank you. We have a minute or two for questions. So, any burning questions? Okay. All right. Thank you so much. I just want to draw the audience attention to one poster associated with this session by Louisa. Some of you might have engaged with it during the Monday presentation. So, yes. Our next speaker is Alexandra, and she will give us a talk on an overview of post-fire regeneration strategies in the Cerrado.
Thank you very much. It's also very nice to be back here, and mostly because I'm talking after my students, and I still have one student to talk. They are coming back to the Savanna meeting, so I'm also very happy to be with my group here. So today, last year I tried to convince you to dig, right? So I was talking to you how important resprouting was. But today, I'm going to talk to you about where do plants come from when we have a fire. So are they coming from seeds? Are they coming only from buds? And we all know, I don't have to explain this figure, that most of the tropical open ecosystems are disturbance-driven. We've heard a lot about herbivory here. We hear a lot about fire as well. And because of that, plants usually have fire and herbivore-related traits. I'm going to talk about a system that is fire-driven mostly because we don't have the herbivores. So we know that the lack of fire will affect negatively vegetation, as Cassie already showed you before. So this is a major figure of how usually plants in savanna regenerate after fire. So we have fire consuming mostly of the above ground biomass. They resprout. They can resprout from above and below ground. Usually they, in some cases like in Brazil, they flower a lot after fire, and then until the next fire comes, it begins all over again and all over again. But sometimes, eventually, we can find some seedlings. Maybe here in Africa you find this more frequently than we do, but all the time I go to the field, I'm looking for seedlings in the rainy season, and I'm not finding them. However, this year I could find some of them, so I was very surprised to find them. But yes, germination is also occurring, it has to occur, but we don't know very well how it does. How is the mechanism behind that? So usually when we talk about regeneration strategies, we divide plants in seeders and resprouters. So seeders usually have the adult plants killed by fire. Population to persist will rely on germination from a viable seed bank, and usually the cycle of the plant is within the fire event. So you need plants to produce seeds to refill the seed bank so plants can germinate in after the next fire. On the other hand, resprouters will resprout after fire, and they will rely on a viable bud bank, which can be above ground and below ground. So plants must need to have some reserves to be able to build up above ground biomass after the removal by fire, and we can have multiple cohorts. So these strategies have been studied a lot in different flammable systems, mostly in the Mediterranean systems. So we know that in some systems it will depend if fire frequency or intensity is higher or lower, so we're going to have more seeders or more resprouters. But I want to call your attention to how fire severity frequency can affect how plants will resprout after fire events, and how it also affects the germination, the post-fire germination. So, as I told you, I will talk about cerrado plants. Cassie and Marco already made a very good introduction of how this vegetation is. I want to call your attention, we'll talk about a very mesic savanna in central that is distributed from north central to the southeastern part of Brazil, and most of the plants have fire-related traits because we don't have mega herbivores since at least 10,000 years in the area. So this study from Pilon, very nice, tried to organize plants according to the regeneration strategies. So I will use this model only to guide us through what I want to say. I will begin with the cedars. And we know we have very few cedars in our system. And it is related to the high fire frequency, so plants, maybe we can think plants don't have time to produce seeds, but we know that after fire they produce a lot of seeds, but very few are obligate seeders. We have very few annuals in the system, and very few only come back through germination. But I will take the opportunity to talk a little bit about post-fire germination, because as I told you, it's very difficult to find seedlings. And one of the reasons may be because we have, as you can see here, very, very few seeds in the seed bank. And I'm talking here about a place that was excluded from fire for a long time. If you go to high-frequent sites, it's even lower than that. It's very difficult to find seeds in the seed bank. And also, after fire in these places, we find very little amount of plants germinating. And when we find seeds in the seed bank, and the native ones, most of them in one of the sites we have are composed of annual grass. So we don't have a very high diversity of seeds in this seed bank as well. And maybe it's because we couldn't make seeds to germinate, so we went to the field, collect seeds before fire in the seed bank. Then we went after fire and resampled again, and for our surprise, we could see that fire stimulated seeds to germinate from the seed bank, but it's still, the amount is not very high. But seeds are there. They can be stimulated after fire, but even though it's very low percentage in relation to resprouting. And one of the reasons may be because since they produce a lot of seeds, we know because we see the flowering and we see the seed production. One main reason that maybe we don't find many seedlings because there is a lot of seed predation by ants. Ants also take the seeds, birds, and other insects. And also we have very low quality of seeds in the Cerrado, so a lot of them have high percentage of empty seeds or non-viable seeds. And also we have a lot of dormancy in the system, so savannas are known for having high dormancy. In our sites we have a lot of physical dormancy and physiological dormancy in the seeds. One good thing about the physical dormancy of seeds is that these are the ones that resist the most, again, high temperatures of fire. So we still are trying to understand the mechanism behind seed germination after fire. We think we are getting there, but we still don't have a very good model to explain that. Resprouters are much easier. So when I talk about the resprouters, I'm talking about all of these strategies that Pilon described. So I'm talking about grasses resprouting after fire. They have buds protected by the leaves. I'm talking about the ones that really, that after fire start flowering very fast. I'm talking about the ones that can spread clonally. And I'm talking about resprouting generally from above and below ground. So we know that most of the species will resprout from basal and below ground buds, showing the importance of these underground structures in the system. And even when we look only at woody species, we see that after a very severe fire, most of them will sprout from the base and below ground. If you take a look at the herbaceous layer after a regular fire, most of them will also resprout from below and basal buds. And we know this has a very tight relationship with fire frequency and severity. So when we have fires of medium and high intensity, maybe if the plant has buds located at the basal and below ground, they will be able to resprout, because maybe the buds above ground are not well protected. This is an example of Fabaceae, Hymenaea styginocarpa from Cerrado. It's a tree, has buds all over, and it's after a severe, low, medium severe fire, it was resprouting from the base. So we also have sometimes fires of high intensity of plants or plants that don't have above ground bud protection. And these plants will rely only on a below ground resprouting. So to do that, they need to have below ground organs with buds that will just let them then resprout after fire. And these plants we also find among, these are most of the forbs, they are all here, and some shrubs and also some trees will do this. And because of this, these plants we rely, as I told you, we rely on these below ground structures, and one of them is this Chilopodium, which is a woody structure bearing a lot of buds, and it's really highly related with fire frequent sites. So we know that when we have fire exclusion, these are the first structures to disappear in the systems, and plants will not be able to resprout from below ground anymore. So we have this scheme here that are showing how plants deal with fire severity where buds are. I told you about how plants deal with when fires are of high frequency or low frequency and they have no above ground protection, and they rely on this kind of resprouting. So this is very important that these plants have either these buds protected by some structure of the plants like the leaves, or they are very well protected by the soil. But we also have plants that are able to resprout above ground, like most of the trees and the shrubs. So they will rely mostly on this bud protection. So when we have fires of low or medium intensity or severity, and these buds are very well protected, this can occur. So you can have plants not having to start all over again from below and trying to escape the fire trap. They can just start resprouting from above ground, which is much better for this plant. So these plants then will rely on the bud level of protection that Marco was showing you. So here you can see that some plants have buds that are not protected at all, But then we have plants that have their buds not only protected by bark, but we are finding a lot of plants that have buds protected by trichomes. We still don't understand very well the role of trichomes because it's not only above ground. Sometimes we found also buds that are on the base of the plant protected by trichomes, but they are also, we believe they also function as bud protection. And we also know that bark production is very important. We know that savanna species in our system produce bark faster than forest species. And bark is not only important for bud protection, but also very important for the cambium protection of the plant. So just to sum up what I wanted to tell you, I hope it was not very fast and you could understand everything. We have these two major post-fire strategies in fire-driven systems. Plants can either germinate after fire or resprout. I'm calling the attention that it's not only resprouting that is important, but where they are resprouting from is also very important, and relating this to fire severity and frequency. So cedars and germination play a minor role in our system. We don't know what triggers, like this year I know that something triggered germination in the system. I think it was because rain was a little bit later this year, but we are not sure about that. So we're still trying to figure out what, it's very complex what triggers germination after fire in our plants. Resprouting is the main regeneration strategy, not only from above ground, but also from basal and below ground buds. So it's really important for you to dig as well and trying to understand how plants are resprouting and behaving after fire. And most of the plants we have this basal and below ground buds. And I want to introduce you what we've been talking a lot in our lab about the Highlander species, and they are mostly trees. I think it's nearly impossible to kill a savanna tree in our system because either they will resprout from above ground if they have a very good bud protection, or if it's a very severe fire and they are top killed and they have basal and below ground buds, they will resprout from below ground. So we have species that can resprout from everywhere. So it just depends on how fire severity will be in the system. And with that, I would like to thank you. I'd like to thank all my co-authors. They are all my PhD students and postdocs, and they are very keen digging plants, doing germination trials, and sometimes assuming the crazy ideas I have to do, implement in the field. And I thank you all for your attention. Thank you.
Thank you, Alexandra. We have 1 minute for a question. Okay, very quiet crowd. Our next speaker is Nikki. Okay, there she is, and she'll speak on the effects of long-term frequent fire on soil and plant nutrient concentration in the Afro-Montana grassland.
Morning, everyone. Yes, my name is Nikki. And today I'm going to be speaking on the results of some of our research on the effect of fire on soil and plant nutrient concentration in Afro-Montane grasslands. So we've heard a lot about savanna systems, but these Afro-Montane grasslands are also ancient grassy biomes, but they are grass-dominated. They occur at higher elevations along the eastern escarpment of the African continent, extending right down to the Drakensberg here in South Africa. They are biodiverse, and they have high levels of endemism, and they contribute vital ecosystem services like water supply, erosion control, carbon storage, nutrient cycling, and importantly in the South African context, they contribute significantly to food security and the local economy. But despite their intrinsic value, only 2% of our temperate grasslands are formally protected in South Africa. They are considered critically endangered, and they are hugely under threat from climate change, land use intensification, and afforestation. A key driver of these grasslands is fire. Prescribed fire is used as a huge management tool in these grasslands, but there's quite a significant amount of debate surrounding prescribed fire, especially because of the negative effects of burning on atmospheric carbon dioxide. We know, though, from recent research that these grasslands continue to sequester carbon and nitrogen, and that this is facilitated by fire. So it's really important that we understand how fire affects not only carbon and nitrogen, but other ecosystem nutrients in these grasslands. So for this talk, I'd like to address three main aspects. Firstly, the effect on soil. So we expect frequent fire to differentially affect nutrients, carbon, nitrogen, phosphorus, and potassium, aka C, N, P, and K. We know that significant amounts of carbon and nitrogen are lost from volatilization, but also that phosphorus and potassium is deposited in mineral forms after fire through the ash or charred organic matter. Small amounts of nitrogen and phosphorus can be deposited through mineralization of heating of the topsoil, but also this ash can be eroded, especially through wind erosion, so you can get losses of nutrients at the same time. We expect the effect to be influenced by season of burn, especially after autumn burns, where we get greater losses, which occur from the bare soil. The second aspect is the plant picture, and we expected that plant nitrogen, phosphorus, and potassium would co-vary with the soil elemental concentrations. And finally, the functional type aspect, we believed that grasses and forbs would differ in their nutrient response to fire and soil nutrients. Our study took place at the Brotherton Burning Experiment, which is located in the Drakensberg on the border of Lesotho. The soils are typical of that region. They're highly weathered, they're basalt derived, and the vegetation is diverse and dominated by C4 grasses, particularly Themida. But the biodiversity is contributed mostly by the forb component. This experiment was established in 1980 to have a look at the effects of fire frequency and burning season on plant community dynamics. It's a replicated randomized block design, and the treatments are mixtures of fire frequency and season of burn. We chose 5 of the fire treatments that had been maintained since 1980, the annual autumn and spring burns, the biennial autumn and spring, and the infrequent burn. The autumn burns take place after the first significant frost, usually in May, and the spring burns after the first major rainfall, usually around October. So we collected soils to a depth of 15 centimeters, and those samples were pooled and analyzed for carbon, nitrogen, phosphorus, and potassium. And biomass was collected at the height of the growing season. The green material was separated by functional type, and they were analyzed for N, P, and K. So, to the results. We did find, in the soils, that frequent fire has a significant effect on soil nutrient concentrations, but that the importance of season is dependent on the frequency of fire and the actual nutrient in question. So if you have a look at this figure, the nutrient is on the y-axis and treatment on the x-axis. That's the annual autumn and spring, the biennial autumn and spring, and the infrequent burn. So carbon content of the soil increased with fire frequency. In contrast, the soil nitrogen and phosphorus were remarkably resistant to fire regime, except in the annual burn, where you see a significant increase in the spring burn in those nutrients. Potassium, in contrast, didn't respond really to frequency, but showed significant increases with spring burns, both the annual and the biennial spring burn. So why is this happening? Well, we know that frequent fires are a great promoter of plant productivity in these grasslands, both above ground biomass, but also probably more importantly in increasing root development. And carbon, nitrogen, and phosphorus are a huge component of that organic material that is produced. So what we think is happening is in spring, we're seeing a reflection of those organic inputs, probably mostly through the roots. And in comparison, you get these autumn burns where you're seeing the negative effects of fire. What happens is you have ash lying on the surface for an appreciable amount of time. Strong winds come up at the end of winter and early spring, and a lot of that ash is eroded by the wind. Potassium is different in that it's returned to the soil in a mineral form. It doesn't have a sort of organic component to it. And so what we're seeing here in spring is the mineral potassium that is being returned to the soil. To the plant question, we expected plant nutrients to co-vary with the soil nutrients, and that did not happen. Here in this figure, we've got grass nitrogen and forb nitrogen against the fire regime treatments, with just a reminder of what happened in the soil on the left. And you can see that there is no significant response in either the grass or the forbs. But if we have a look over here, this figure far to the right, if soil nitrogen increases, we do see that grasses have the ability to increase their nitrogen. And that for us was a bit of a question. So we looked more closely at it. Why are we not seeing that increase that is seen in the soil nitrogen reflected in grass or forb nitrogen? And we think it has something to do with this really neat paper that was published by Clint Carbett from KZN Wildlife, where he showed that in these grasslands, although they are intrinsically nutrient rich, they are functionally nutrient limited. And that is because of seasonal temperature limitation of microbial activity. So what we think is happening is most of this organic nitrogen, well, this nitrogen that we're measuring in the soil is actually still in an organic form. And it's not available to the plants for uptake. So you don't see it reflected in plant nitrogen content. We see a very similar picture with the phosphorus, where there is no significant response to fire treatment with phosphorus. And we suggest it's the same thing happening. It's almost like a bottleneck to nutrient turnover that happens as a result of the microbial temperature limitation. Finally, potassium, you do see a response. And again, I think it's because we're having the potassium is returned to the soil in a mineral form, which is immediately available for plant uptake. And so you can see a plant potassium response. What's interesting is that we don't see an increase in grass or forb K with increasing soil K. And I would imagine it's because there is no, sorry, I was distracted by the 5 minutes. K is not limiting in the system. So we're actually seeing the top of the curve rather than increasing response with increasing K. So finally, we looked at the nutrient concentration between the functional types, and we put together a PCA to understand the relationship between functional type and ecosystem nutrients. And this looks like a pretty complicated picture, so I just wanted to tease it out a little bit and as quickly as I can. We saw that the soil and plant nutrients separate out along the two PCA axes, which confirms the poor correlation between soil nutrients and the plant nutrients. When we included plant nutrients by treatment plot and functional type, we got this, what appears to be a random scatter, but we can still see that regardless of plant functional type, the spring burns were associated with above average soil nutrient content, while the autumn burns with below average soil nutrient content. And the infrequent burns, they sort of occupied a similar space to the biennial autumn and spring burns. When we included the grasses and the forbs, we see that they separated out along the plant nutrient axis. The grasses associated with lower plant nutrient content and the forbs with higher plant nutrient content. And we know that forbs in this system have underground storage organs, and I would imagine that those facilitate a more constant supply of nutrients to the above ground biomass. What is a bit odd is that we have 3 plots where the plant, the grass and the forb content is pretty similar, and it's not one particular treatment, so we still need to look a bit more closely at that. So to summarize very quickly, fire frequency and season of burn did differentially affect soil nutrients. The plant nutrients did not co-vary with the soil nutrients, and the biomass relationship to plant nutrient variables could be grouped by plant functional group with a distinct clustering between the grasses and the forbs. And to conclude, the soil nutrient response to fire regime reflects the net effect of nutrient returns. So that would be increased productivity, returns in the ash, mineralization, against losses due to volatilization and erosion. The grass and the forb communities occupy different nutrient niches and have different nutrient strategies. And we see a grassland that is functionally dependent on fire, but actually resilient against a whole range of fire regimes, which probably reflects the historic fire frequency. Our next steps, at the moment, we are busy with a paper that looks at soil phosphorus pools, responses to fire. And we also have a project looking at the response to fire along an altitudinal gradient here in KZN. I think an important question is, if climate change brings warming, will that alter our response? Will microbes start being able to be active the whole year round rather than being seasonally limited? And that's our story. Thank you very much.
Thank you, Nikki. We have 1 minute for a question. We have a hand there.
Fascinating talk. Just curious about the prevailing fire regime. What is the surrounding vegetation fire regime?
So at the moment, this area is mostly managed by KZN Wildlife, and they implement a biennial fire. So every second year, they block burn, and they try and maintain the grassland as a grassland, but I think also moving towards trying to implement more heterogeneous burns, because you do tend to lose some of the rarer forbs with too frequent burning. I think historically, you wouldn't have had lightning occurring every year or every 2 years in a sort of regimental fashion. So I think the move is towards trying to mimic historical fire frequencies to a greater extent.
Is that another hand? Oh.
I was just wondering if you couldn't do, besides, I think you did total nitrogen. Maybe if you did nitrate and ammonium, you'd see more of a response in your soils. You would
Sorry,
see
if
something.
we did nitrogen,
So you didn't find a response of your plants to changes in soil nitrogen, right? But maybe that's because you did total nitrogen.
Exactly, yeah.
So maybe do the inorganic, which is more available.
So our total nitrogen obviously reflects both the inorganic and the organic. In these soils, there is very little inorganic nitrogen. It's very low. And I think having looked at these results now, it would be worth going back to look at nitrate N and ammonia in the soil, because that, I think, would prove the hypothesis that we are seeing a limited turnover from organic to plant available nitrogen.
Okay, thank you, Nikki. Our next speaker is Tiffany Play, and she's going to give us a 5 minutes titled Nutrient Addition, Fire and Grass Competition Affects Biological Nitrogen Fixation in Vachelia.
Good morning. Today I will be talking about nitrogen fixation in a common woody encroaching species. Woody plant encroachment is resulting in the transformation of millions of hectares of land from these grassy open systems to closed canopy systems with cascading negative consequences. The species that are implicated in this directional increase are usually from very select genre, specifically Vachelia and Senegalia, and many of them are capable of biologically fixing nitrogen, many but not all. This ability to biologically fix nitrogen has obvious implications for the terrestrial nitrogen cycle, but it also confers a distinct competitive advantage to these species, particularly in the seedling stage when competition between trees and grasses is extreme. Vachelia cibriana is a common encroaching species that occurs in the mesic savannas of southern Africa, such as those that our previous speaker just mentioned. These savannas are nutrient poor, and so the ability to biologically fix nitrogen has large implications. For this reason, lots of recent research has focused on nitrogen fixation at the seedling stage. This study looked at nitrogen fixation, or actually nodule formation, in Vachelia cibriana seedlings across a gradient of soil moisture content, and they found that at the low soil moisture content, so in droughted conditions, fewer but larger nodules were formed. Now, nodulation is an important indicator of nitrogen fixation because these are the actual masses on the root surface in which N2 fixing bacteria are housed. Another study has shown that competition can affect nitrogen fixation in cibriana seedlings. In this study, Vachelia cibriana seedlings were grown with competition from an invasive alien shrub, Chromalina, and they found that atmospheric derived nitrogen was higher in plant tissue grown with competition, showing that nitrogen fixation was higher. So for this study, we wanted to look at the independent and interactive effects of nitrogen, of various drivers in savannas on nitrogen fixation in seedlings of Vachelia cibriana. We did this in a greenhouse experiment, and we added nutrients, we simulated fire, and we clipped the grasses to simulate grazing. We measured growth and survival and a range of semi-useful below and above ground traits. And importantly for this study, we looked at the number and weight of nodules that were produced, as well as the rate of biological nitrogen fixation as given by the acetylene reduction assay, which measures the rate of reduction of acetylene gas into ethylene in the presence of nitrogenase. As expected, we found that bigger trees do fix more nitrogen, so there was a significant positive relationship between the rate of nitrogen fixation and plant size. But interestingly, this was also found for both the mass and the total number of nodules. This confirms nodulation for the species, but it also shows a relatively high investment in nodules per unit plant mass, which is good for plants of that size. We found significant independent effects of fertilizer and fire. So the no fertilizer treatments and the fire application treatments produced the highest rates of nitrogen fixation, which we did expect as compensatory effects for the low nutrient status that these treatments give rise to. We did, however, find no significant effects of the grazing treatment, which may indicate that above ground grass competition was did not have an important effect on nitrogen fixation rates. However, total grass biomass did significantly negatively affect the rate of fixation as we expected, and it also resulted in significant mortality of the tree seedlings. So we conclude that actually grass competition is quite a negative, has a negative impact on fixation in these species, but importantly that nitrogen fixation is facultative in these in Vachellia cebriana, so it's highly inducible in response to these drivers. Grass competition negatively affects nitrogen fixation, and it's important to note that while you might see a higher induced effect at lower grass biomass, this might be negative as this competition actually increases. And this suggests that there's a threshold of grass biomass below which nitrogen fixation confers a distinct advantage to the species. Thank you.
Thank you, Tiffany. Our next speaker is Yani, and she's going to give us a 5-minute presentation on the effects of fire frequency on recruitment attributes.
Hello, everybody. Like she said, my name is Yani Stein, and today I will be presenting my master's study and the preliminary results we have uncovered so far. The title of my master's study is The Effect of Fire Frequency on Recruitment Attributes in a Semi-Arid Savanna. So as we all know, fire is an important driver of savanna vegetation function and structure through its influence on the recruitment and mortality of different functional groups and species. However, there is a knowledge gap in the literature pertaining to how fire influences the soil seed bank of these savanna systems, and that is what I aimed to address in my master's study. My master's study has 3 main components, the first of which is to try and determine the effect of different fire frequencies on soil seed bank size and composition. The 3 fire frequencies I looked at is annual fire, triennial fire, and fire exclusion. And I wanted to see how they influence soil seed bank size and composition. Then I also had a standard germination trial where I collected seeds from the dominant grass and forb species within my treatments, and I put them to germinate to try and see which species produced viable non-dormant seeds and which ones produced viable dormant seeds. Species that did not germinate well in the standard germination trial were tested for viability with tetrazolium testing. If they had high amounts of viable seeds but did not germinate well in the standard germination trials, they went through to dormancy breaking trials where they were exposed to fire-related dormancy cues. The results we have so far is for the soil seed bank. We saw that alteration in fire frequency does not deplete the soil seed bank, but it does alter the seed bank size and forb-grass ratio of species within the soil seed bank. For example, in the fire exclusion plot, we saw the soil seed bank is dominated with grass species and has a very low proportion of forb species. It is also a very large soil seed bank. In contrast, in the triennial fire frequency, we found that the soil seed bank size is smaller, but it is much more diverse, and also the forb to grass ratio is much more even. In the annual fire frequency, we found that the soil seed bank size is quite small, and it's also not very diverse. Moving on to the standard germination trials, the general trends we've observed so far is that the grass species tend to germinate much easier than the forbs, and they seem to have fewer dormancy characteristics, whereas many of the forb species have hard seed coats relating to physical dormancy, and we are now testing the species that had high amounts of viable seeds but low germination success. We are putting them through dormancy trials where we expose them to heat shocks and smoke water to simulate a fire event and see if we can break the dormancy of those seeds. That is what I've got for you. Thank you for listening.
Thank you. Great job, Yanni. We have 1 minute for a quick question. Oh, they just.
I hope I didn't miss it, but what do you think the longevity of the seed banks are?
The what?
The longevity.
Of the soil seed banks?
Yeah.
I think being closer to the natural fire return cycle is best for the soil seed bank, simply because it supports a greater diversity of species, and also because it allows for the forb to grass ratio to be most equal. I think that's going to allow for the longest continuation of the soil seed bank.
Cool, thanks, Yanni. Off to our last speaker, Savanna. He's going to discuss flammability-related traits distribution according to fire frequencies and time in the Cerrado. Take it away.
Morning, everyone. I'm Wagner Zanzarini, and I'm a PJ student from São Paulo State University. And today I'm going to talk about how fire frequency and time since last fire affect flammability-related traits in open savannas. So we know that different fire regimes are responsible for assembling functionally diverse plant communities, influencing the way that fire spreads across landscapes. And this can affect the system's flammability. So we may expect that changes in fire frequency, together with the time since last fire, which are fire regimes characteristics, could lead to changes in flammability-related traits of species. And this can help us to understand fire behavior and to make future decisions on fire management techniques. So our main question regarding to such ideas was to evaluate what changes do we observe in flammability-related traits in areas with different fire frequencies and different times since last fire. So we selected areas of open savannas in the Cerrado, Central Brazil, choosing 4 areas with different fire frequencies and time since last fire in an interval of 21 years. So the first area we have was a high fire frequency area recently burned in 2019. And the second area was also a high fire frequency, burned just in 2011. While the third area was classified as a low fire frequency area, burned in 2017. And the last area was also a low fire frequency area, but burned just in 2001. Having more than 20 years of fire exclusion. So in the 4 areas, we established 30 1-per-1 plots in the end of the dry season, and selected species representing more than 80% of the entire plant community abundance of graminoids, forbs, and small shrubs. And after, we accessed the following flammability-related traits in each species, in individual species. So we accessed the plant dead biomass, the plant moisture content, and we also calculated the burn rating using an equipment to measure flammability components in the field. So in relation to our findings, we could observe that the high fire frequency area recently burned and the area excluded from fire for more than 20 years were completely different from each other. And when we accessed the PCA axis, we can see this clear separation between these 2 areas. And we can also see that the area with high fire frequency recently burned have an association with burn rate, and the other area an association with dead biomass amount. And when we look to the burn rate, we can see how the first area, the high fire frequency area, plants burned fast, and in the area with low fire frequency, plants burned slowly. And these can be associated with the amounts of dead biomass, because in the first area, plants exhibit over 50% of dead biomass, and this can be considered aerated foods contributing to the rapid spread of fire. And in the area excluded from fire, plants exhibit over 80% of dead biomass, and this can be considered condensed foods, decreasing the spread of fire. And in relation to moisture content, we can observe that these two areas, the high fire frequency recently burned and the area excluded from fire, exhibit high percentage of moisture content. And in the first area, this high moisture content can be attributed to the presence of forbs and shrubs, for example, the high presence of them in the ground layer. And such species are more humid than grasses, for example. And while in the last area, the humid plants can be associated with the presence of a high dominance presence of graminoids in the ground, which are accumulating that biomass over 20 years of fire exclusion, and this material can be dense and humid. So to conclude, we can say that fire frequency and time since last fire led to changes in flammability-related traits of species. And in all areas, plants burned different, and when it comes to the areas with high and recent fire occurrence, plants burned faster in comparison to the area excluded from fire. And the exclusion of fire promoted an increase in the amount of dead biomass of ingress to socks, decreasing the spread of fire in plant individuals, thus modifying the fire behavior. And our findings contribute with information that might be useful for decisions regarding fire management. So thank you very much for your attention.
Thank you, Savanna. You have used up all of your time. I just want to thank all the speakers for their insightful presentation, and I also want to apologize for butchering some of your names. I just also want to encourage the audience to engage with the speakers during breaks, most especially the ones that went over time. Just hunt them down and ask your burning questions. So a round of applause, please, for all of our speakers. Just a few reminders before we go to tea. Was asked to remind everyone to please fill in the feedback form for us, and for the poster presenters to take down their posters during lunch. Thank you, everyone.